neurolegin-4 in quite some detail as well, biochemically and cell biologically, and what I can show you in form of a summary again, that we found that it operates by the very same principles as neurolegin-2 and distinct from neurolegin-1 and neurolegin-3, and that it also binds the scaffold proteins colibistin and gefferin, and that in vitro conditions or upon exogenous expression, for example, in fibroblasts, can organize inhibitory postsynaptic protein scaffold as was seen for neurolegin-2. But this hasn't really helped us much in understanding why neurolegin-4 is so intrinsically and fascinatingly involved in autism spectrum disorders. And one reason for that is that neurolegin-4 is among the neuroligins the least abundant. It's about 10 times less abundant than any other neuroligin, which has caused us major problems in really understanding what this molecule does. And the first insight came from the retina of all places, and you might now with due right be disappointed, because the retina will not tell us much about autism spectrum disorders, but it tells us something about what these molecules are doing, because neurolegin-4 is apparently very strongly expressed in the retina. What you see here is a cross-section through the retina, and for those of you who don't know much about the retina, it's sufficient to understand that we have two synaptic layers in the retina, the outer plexiform layer and the inner plexiform layer, in which synapses between cells in the retina are formed. And this inner plexiform layer consists of synapses that are formed between the bipolar cells here and this inner nuclear layer in the ganglion cells down here. And this inner plexiform layer contains two major types of synapses: glutamatergic synapses formed between these already mentioned bipolar cells and the ganglion cells, and then inhibitory synapses formed by amacrine cells upon different postsynaptic targets. And both of these synapses can not necessarily be distinguished by standard markers, but if you use the appropriate markers, you can do that. For example, as illustrated over here, I'm showing you a staining of the inner plexiform layer, stained for neurolegin-4, and glycine receptors here on the left-hand side, and of neurolegin-4 and PSD95 on the right-hand side. And what you see immediately is that there's no coincidence of labeling between neurolegin-4 and PSD95, indicating that neurolegin-4 is not at glutamatergic synapses, for which this molecule here is a marker, but there is a high degree of co-localization between neurolegin-4 and glycine receptors, indicating that neurolegin-4 in the retina is localized to a subset of glycinergic synapses. And this has been studied by us in quite some detail, also in different brain regions. And the gist of all these data is that neurolegin-4, like neurolegin-2, is localized very specifically to GABAergic synapses, but not to all GABAergic synapses. We studied the retina in a little bit more detail, and to our disappointment, the loss of neurolegin-4 has very little effect on retinal function. What I'm showing you here is basically all we could find. Here on the top left, I'm showing you immunostainings of the retina of wild-type and neurolegin-4 knockout mice. For different glycine receptor subunits, we quantified these kinds of staining with various methods. The quantifications are shown here. There's hardly any change that we see, apart from a slight reduction in the level of glycine receptor alpha-1 subunit. And this is predicted to have slight consequences of glycinergic synaptic transmission, delaying the decay time course of glycinergic synaptic transmission. This is exactly what we find. What you see here are postsynaptic measurements of ganglion cells stimulated by glycinergic input. You see a wild-type cell response and a knockout cell response. And the only difference we see is very subtle, is that the knockout responses have a slightly extended decay time course, indicating this partial loss of glycine receptor alpha-1 subunit. There are a few other small changes in the retina, not very informative, apart from the notion that these molecules play a role at inhibitory synapses, but they are not totally required for proper function of these synapses. Another piece of evidence that I would like to share with you, and that supports the notion that neurolegin-2 and neurolegin-4 operate by the same principles and are probably functionally redundant, is the image shown here, where we have a wild-type and a neurolegin-2 knockout retina stained for neurolegin-4, and you see that neurolegin-4 labeling increases as neurolegin-2 is knocked out, indicating that these two proteins cooperate at inhibitory synapses and can replace each other. And we have corresponding data for the neuroligin 4 knockout as well. So what I would like you to remember from these data is that neuroligin 4 is most likely an inhibitory synapse neuroligin that operates like neuroligin 2 in recruiting receptors for GABA and glycine to these synapses. But all this really hasn't helped us much in understanding why neuroligin 4 plays a role in autism and why its mutation in human patients leads to such interesting yet severe changes in behavior. And one of the reasons why we had such difficulties was that we had very, very great difficulties in detecting the protein in other parts of the brain in the first place. And only recently were we able to generate antibodies that helped in solving this problem, at least to a certain degree. And what I'm showing you here are rather new immunostainings of a mouse brain labeled for neuroligin 4. And you see that in the brainstem, for example, there's very strong neuroligin 4 staining, and here it is very, very nicely localized to inhibitory synapses. But in the rest of the brain, particularly in the areas of the brain that might be relevant to autism spectrum disorders like the hippocampus or in the cortex, the staining is very, very dim. Yet specific, as illustrated down here, for the somatosensory cortex, this is a wild-type section and this is a neuroligin 4 knockout section. You see there is specific staining in the somatosensory cortex, particularly in the layer 4 of the cortex, and you can even discern with some biofantasy that individual barrels of the somatosensory cortex are stained. Over here you see an image of a wild-type and a knockout hippocampus stained for neuroligin 4, and you see that the neuropil in the hippocampus is positive for neuroligin 4, weakly positive, and there's particularly strong staining in this area here of the stratum lacunoso moleculare, to which we can return in the discussion period of the talk if you want. Now with this information we set out to do experiments in the somatosensory cortex and in the hippocampus, and to our dismay we were really not able to find any clearly measurable synaptic defect in these networks by, for example, patch clamp recordings from cells here in the CA1 region or from cells here in the layer 4 of the cortex. So we basically regrouped, thought, and assumed that maybe the defects that we see at these synapses, that we can possibly see at these synapses, are so minor that they are very hard to measure at the single synapse level, and therefore studied certain aspects of network activity in these populations of neurons that could help us to indirectly read out changes in inhibitory synapse function. And one example I would like to show you with this image, where we measured in the hippocampus CA3 region, this is over here, with extracellular electrodes, oscillatory activity that is known to depend on the function of inhibitory interneurons that are in a feedback loop and that drive oscillations in these hippocampal CA3 cells, an oscillation that is called gamma oscillation. These oscillations can be triggered in the CA3 region by adding an agent called canic acid, which stimulates glutamate receptors, and if you do this in a wild-type slice and measure, you find really strong oscillatory activity in such wild-type slices. And really, to our fascination, in the knockouts, we found hardly any such oscillations. This is quantified over here, where you see the power of the oscillations as a function of the frequency. Very strong gamma band oscillations in the range of 40 hertz in the wild type, and very much less in the knockout. And this is really the first tractable change at the level of an interesting protein, of an interesting brain region in these neuroligin 4 knockouts. And we are now exploring this further. This may well be involved in modifying complex activities, even learning activities, and future studies will have to help us understand how neuroligin 4 is involved in guiding or in driving these oscillations. It's a bit of an anticlimax, but I would like to summarize this part of the talk by saying that in retina, brainstem, spinal cord, globus pallidus, and superior and inferior colliculi, neuroligin 4 is localized to a subset of inhibitory synapses. Glycinergic transmission and network function are slightly perturbed in the neuroligin 4 knockout retina. I showed you these data. In hippocampus, neuroligin 4 is diffusely localized to the synaptic neuropil. There are enriched levels in the stratum lacunoso moleculare. And the barrel cortex is localized to both glutamatergic and GABAergic synapses, particularly in layer 4. And we think that functional synaptic consequences of Neurogen 4 loss are very subtle, and this is already indicated by the subtle changes at the synaptic level and the more profound changes in network activity like these oscillations. So we think we have to explore network characteristics further and maybe step away from synaptic recordings in order to see what the consequences of Neurogen 4 loss are. But I would like to end with a data set that is probably more fascinating to you and certainly more revealing in terms of the role of these molecules in autism. I told you that Neurogen 4 loss of function mutations in humans cause autism spectrum disorders. And we had generated Neurogen 4 knockout mice, which basically therefore are a valid model in terms of construct validity for autism spectrum disorders because we generated mice with a genetic lesion that is known to cause a disease in human patients. And we set out to test whether these mice show any behavioral characteristics that would be reminiscent of autism spectrum disorders. In preparation for these experiments, we characterized these mice behaviorally in very much detail. We showed that they have normal motor characteristics, they have normal motor coordination, they have normal anxiety, normal vision, normal smell, normal taste, normal hearing, and learning characteristics. And we then started to study these mice in behavioral paradigms that are actually more relevant for social behavior. This can be done in mice quite nicely because mice are social animals. So for example, if you put mice together in pairs, they start to sniff each other in awkward ways, I must admit. But nevertheless, and you can score these interactions between, for example, a pair of wild-type mice and a pair of knockout mice. And what we saw was that wild-type mice spent more time in contact than knockouts, indicating some aspect of change in the social behavior domain. Probably more informative essay is this one over here, in which you put a mouse into a tripartite chamber and give her the option to choose to spend time over here with a conspecific or over here with an empty cage. And each normal wild-type mouse will spend more time over here with a conspecific mouse rather than over here with an empty cage, and this preference can be read out very robustly in these kinds of assays. And what we saw was that this preference, which is really clearly discernible in wild-type mice, is totally gone in knockouts. They seem to be equally interested in the empty cage, indicating that there is a defect in social approach and maybe in social interaction in these mice that is kind of reminiscent of autism spectrum disorders in humans. And the final data set that I would like to share with you concerns language in mice. Of course, mice can't speak, but mice communicate with ultrasonic calls. There are typically two types of calls. Calls in the lower 22 kilohertz range are emitted upon aversive stimulation, for example, when pups are removed from their parents or when these animals, when adult animals express anxiety or fear. And then there is another type of call which is emitted at very much higher frequencies, also has interesting frequency modulation characteristics as illustrated up here. And these calls are emitted by mice upon positive emotional stimulation. And a standard setting in which these assays are done is that you put a male mouse into a cage with a female mouse in heat. And as is the case with humans, male mice start to sing when they are together with a female mouse that they like. And this kind of behavior can be assessed very nicely in such settings. And this is illustrated in this next slide, where you see on the top, or sorry, in the middle here, calls emitted by a wild-type mouse upon putting it into a cage with a female in estrus. And down here, a Neurogen 4 knockout mouse. You see here the calls in black. You see many less calls in the knockout, which can be read out quantitatively, and also the male mice lacking Neurogen 4 take longer to start emitting the first call. So not only do these Neurogen 4 knockout mice show behavioral defects in social approach, they also show behavioral defects in communication, in this case in ultrasonic communication. So to summarize this last part on autism, I would like to state that autism is mainly a genetic disorder, and that mutations of single synapse genes, in our case here neurolegin 4, can cause monogenic heritable forms of autism. There's a major autism susceptibility pathway, as far as we see it, involving presynaptic neurexins, postsynaptic neuroligins, and then postsynaptic scaffold proteins of the Schenck family. And there are more and more proteins, or more and more genes being found in rare forms of monogenic heritable autism, and many of these kind of link up to these 4 types of proteins. So based on these data, we would argue that autism may be a synapse disorder, a synaptopathy. I showed you that neurolegin 4 knockouts show 2 typical signs of autism-like behavior that mimic 2 key symptoms in patients: aberrant social interaction, aberrant vocal communication, and therefore we think we have established face validity of this model and hope that the neurolegin 4 knockout model might in the future be useful to establish diagnostic and therapeutic strategies for autism spectrum disorders. At the heart of this, we think that there is a deficient inhibitory synaptic transmission in the forebrain of neurolegin 4 deficient patients, probably, and also mice in the future will tell how exactly, or what exactly these changes look like and how they can be modified in order to maybe achieve therapeutic potential. I would like to end by showing you the slide of collaborators. Again, you can read it, but I hope you will also have some time to ask questions. I thank you for your attention.